Hi, so I am from National Institutes of Health, which is located in Bethesda, Rockville, Frederick, and in North Carolina also. And it comes under Department of Health and Human Services. Like DHHS has FDA, CDC, and NIST and many others. And there I am the program director in the program called Epidemiology and Genomics or Genetics program. And our division is called Division of Cancer Control and Population Sciences. And I am in National Cancer Institute. So today I will discuss about metabolomic research at NIH. Since I am from Cancer Institute, so I would like to emphasize about the disease, how serious it is. So either cardiovascular disease or cancer, they are the #1 killer in US. In US, about 4.24 million men and 5.31 million women, they are affected, and death rate is also very high. So our idea is that what we can do by different approaches, either to prevent this disease or treat that or control that. And in the cancer, it develops in different stages. So these stages are shown at different places, and for major cancers like breast, cervix, colon, prostate, lung, it takes 15 to 20 years to develop. Unlike ovarian cancer or pancreatic cancer, where incidence and survival and all that are 2 to 5 years. So in these cancers especially, something we can do at early stages. So if we can identify some markers at early, then we have option either to treat or prevent or do something. Although in our program, we follow up patients also after treatment. So that is also important, but for control, initial stages are more important. NIH has about 6,000 scientists. Its budget has been flat for some time, but still $31 billion are there, which we distribute across the country as well as outside also. And 10% of NIH budget goes to intramural research, which we do at NIH. We support about 4,000 institutions, 300,000 scientists, research and personnel. So what NIH did about 6 years ago, Dr. Zahruni was the director of NIH. So he initiated one program. He says that out of 28 institutes, which are basically based on either organ site or disease. For example, ours is Cancer Institute. One is National Institute of Allergy and Infectious Disease, or kidney disease, eye disease, based on that. But he proposed that we should have certain theme, which is either based on mechanism or such disease, which is of interest of many institutes. So if something is such, which is of interest of 3 or 5 or more institute, then 1.5% of NIH budget will go to that. Initially it was called Roadmap, and now it is called NIH Common Funds. And now 2 years ago, Congress made it mandatory. So it is congressionally mandated budget 1.5%. Whole program is called NIH Common Fund. So since then, we have initiated several projects which are shown here. And I have been involved from the inception in epigenomics, and this one, new one in metabolomics. So that has been there. And what we do, first we develop the resources. Who can do that kind of research, who are doing, and then we ask investigators to submit different proposals. So that's how it works. And these are based on cooperative agreements, which means that once fund is given, then NIH can discuss with the investigator and follow up the progress. If something has to be modified, that also we can do. So that's the whole idea. When it was initiated, then few of us discussed in that, and from NCI, Dina Singer was there, I was there, and then our director and other officials were there. And just 1 month ago, our director, Harold Verma, who is a Nobel Prize winner, he was the director, now he has moved to Cornell. So we decided that what is the common factor or common mechanism which exists in many diseases. So whether it is diabetes, cardiovascular, cancer, or obesity, in that metabolomics was found of great interest. And that's how this Common Fund Metabolomics was started. And what area or program we wanted to support? First of all, standards and protocol, so that we can quantify very well. Sometimes it is targeted, sometimes untargeted, and many metabolites are there. So we were more interested in unidentified, so how to do that. Then informatics tool process, how data is stored or disseminated, and then core understanding of how much and how often individual metabolites vary under normal conditions, especially in clinical samples. When we initiated this project, many people were hesitant that if you eat something, then your metabolomic pattern will change. But when clinical samples longitudinally they were followed after certain interval, then disease-associated peaks, they were very prominent. And lot of epidemiological or clinical studies we have done, so that also indicates, so this has that power. Then improved tools for measuring subcellular localization and changes in metabolite concentration. Thank you. Now we have a working group at NIH in which people from different institutes, they participate, and as I am showing here, Barbara Speholz, and I am from NCI. But all other people are also heavily involved. Question was, why metabolomics? Because it enables simultaneous identification and analysis of multiple metabolites in different kinds of samples. Then we can do global analysis of small molecules and determine their profiling. Only metabolites are such which truly reflects physiological condition or your phenotype. If you are going for genomics, you will see some changes, but they may reflect to functional assays or functional things or not. Then we have to come to RNA or protein, but metabolites has that advantage. Another thing, metabolites, especially disease-associated metabolites, they are very much stable. Mostly in clinical samples, either you collect urine or blood, which we can use. And in our resources also, biospecimen resource, those are the samples we store. So we thought that that is the good one. And metabolomic, okay, this I covered, that it reflects the phenotype. And additional information, how individuals respond to therapy. So prognostic or therapeutic markers for those metabolites we can use. Then integration of biological pathways to understand the biological mechanism and gene-environment interaction or other interactions. And then we can produce metabolomic fingerprint or metabolome for integrating with proteome or other kind of information. Now, before we started that program, we wanted to know how much research we are already doing at NIH. So this is shown like kidney disease. In that institute, about 16% of budget was going for metabolomics, general medicine this much, and cancer also 12%. These were the ones. Mostly this was focused on non-human subject, and some research was on human subject as well. And if you see at the time when we did analysis in terms of funding, this was the dollar amount we were spending. So anytime we make a program or new program at NIH, we analyze how much we are spending and how much more we should put. Then annually also we see how much it is going on. So in funded projects, 115 were the R series, like investigator initiated, R01 grant, R37, R21, all those. And in that, at least one aim was metabolomic oriented. Then cooperative agreement or U series grant, in which several cores are there, so at least one was on metabolomics. And in some grant, metabolomics was a minor component. Most of those projects were for technology development. For clinical implication, there were not much. So we wanted to emphasize how we can use whatever information we have for clinical purposes, or how we can move in that direction. And the way it was started initially, so we put roadmap for metabolomics, and then after that, either for technology development or standard development, at different times, we started putting money on that, whether lipid analysis or others. And then these are the examples of some of the projects which were done at NIH, and they are related with either basic mechanism, cell line model, animal model, or human, whether it is glycomics, lipidomics, so several kinds. And these R21 and R33, that is such mechanism where exploratory ideas are tested. no matter you don't have preliminary data, but we'll support that if resources are there, you have a skill and idea, and based on literature you are proposing that. About investigator-initiated project in which you should have a hypothesis and some data. These were the projects which were done, and we are very much interested in those also where host-virus interaction you study and how much host metabolites are involved. Because in most of the cases, infections are there in our body, those are there, but certain time comes when it triggers to a disease, not all the time. So those were few of the examples. And in that case, time to time we organize some workshop or get recommendation from outside. Similarly, this one was done initially, and then we follow up internationally also what other people are doing. For example, in Australia and England, very big centers are there, and I have visited most of them. Some, like England, which is in where Imperial College London, Jeremy Nicholson is there. So he analyzed samples from SmithKline Beecham or Johnson & Johnson. So they are very much clinically oriented and targeted approach. Other people are doing basic also. So Netherlands, UK, Canada, and total global estimate when we took at that time, it was about $225 million. And this is the distribution in different omics technology at the time we did for genomics, metabolomics, proteomics. So genomics is the number one, and the way we have done in genomics, whole human genome sequence we know. On the same pattern, NIH started epigenome project to know in normal, healthy-looking people or pathologically healthy people, what is the profile of methylation, histone modification, microRNA, chromatin compactation. And now we are proposing that in healthy samples, what is the metabolomic pattern, so that we have some reference point to compare with different diseases. So that is the idea. And that time when we saw the distribution of money for different areas, so technology development or informatics or tool development or metabolomic flux, basic or translation research. So this was in general the pattern. And this kind of analysis we do at NIH because every year NIH get funds, and then we have to propose Congress or justify that this is the area we want to promote, or this is the area we have developed enough and we want to move to some other places. So that's how it is done at NIH. And these are those centers I mentioned, which have all kinds of resources to do different kind of metabolomic profiling or metabolomic things. When we started our NIH program, so the centers shown in green, these are at least those places where some metabolomic project is going on. So at a time, what also we do, sometime if someone comes with good idea, but that person or institute or investigator does not have experience, then we can tell, okay, these are the people, we help them in connecting to those. And time to time, we put outside some money, okay, this is set aside money, people should make project and submit to us, then it is reviewed and funded. So for this program, 110 million for 5 years we committed. One was to develop regional comprehensive resource center. Another one was nomination of mentored or training award. Another one was for doing workshop or training, then technology development, and then one data coordination center. Because as you know, genomics has some data which is used. When proteomics came, it was 10 times more. Metabolomics is further more. So people at different institutes, all institutes do not have that capacity that they can hold that data. And NIH, since it runs by public money, so our idea is that that data or tools developed should be disseminated or exchanged. So for that, we have made this data coordination center. And as you know, we support CR database or NCBI or others also, that way we can do. And time to time, if we have to add something, then through notice we announce to the world that we are asking for that. In our program, since I am from cancer epidemiology and genomics, certain research questions which specifically we wanted to ask, like, can metabolomic profile identify populations at high risk of developing cancer? Or how we integrate informations from different omics technology? Or developing metabolomics-based mathematical models or risk prediction models by which we can tell, okay, you are exposed, some population is exposed to certain thing, who are likely to develop cancer or other diseases. And then metabolomic pathway, response to environmental exposure and individual susceptibility. Because especially in cancer, only 8 to 10% cancer is genetic. Familially from parents you get, but all other is due to environment. So understanding environment, what is affecting which genes and what metabolites are affecting, that is of high priority to NIH. Then identifying and validating metabolomic biomarkers and biological responses to exposure and how early indicators of cancer can be identified. Then study design to measure responses after the treatment, that is also there. And can metabolomic profile identify subtype of cancers? As you know, in breast cancer, ER triple negative or ER positive negative, those are there. Prostate cancer or lung cancer also have 15 or 20 kinds. Whether this profiling can identify that subtyping and how we can integrate with the pathological information. Can unidentified metabolites be used for cancer detection, diagnosis, or prognosis? How can we use metabolomic profiling for personalized medicine? I think 1.5 months ago or 2 months ago, our president announced that personalized or precision medicine, that is of high priority, and $215 million additionally NIH is getting for personalized medicine. As you know, everybody's immunity is different. You give one drug to 10 people, 8 will respond, 2 will not. So since doctor follows those patients longitudinally, so we have to be following the personalized medicine, and in that profiling, whether it is metabolomic, epigenomic, or other kinds of omics along with genomics, that if we integrate, then probably sensitivity and specificity of those markers we can increase. Some examples, in this case metabolomic was done, and these are in fact called metabolomic wide association studies. Populations from China, Japan, UK and USA, they could be identified based on totally metabolomic pattern. This is the website for Common Fund Metabolomics, as I mentioned. So this we time to time update. Not only research opportunities will be there, but different time workshops are organized, and we are associated with Metabolomic Newsletter and Metabolomics Society. In June, mid-June in San Francisco, Society of Metabolomics, they are meeting, so we are organizing one session on epidemiology and metabolomics. And for common funds, as I mentioned, that they will support not only the project, but many times in your research, for your standard, you don't have such metabolites commercially available which you need, because you want to compare against something. So we have dedicated amount and people who will generate unidentified or even identified but not commercially available metabolites, and after we get, they are characterized and distributed so that you can use. It is similar to the TCGA program which we initiated for cancer, that tissues were collected very well characterized. So if you want to do methylation analysis or genomic or SNP analysis or copy number variants, that you can do. At the same time, we want to train new scientists in metabolomics. So on this website, we'll put that which centers are doing those workshops for 1 day, 3 days, or 6 days, and real data also you analyze. We provide laptops to all participants and already loaded with tools, then you do some experiment and analyze. So that way we are promoting that area. Now, SBI and STTR programs are such. These are a small business program at US, and whole government want to support that. In that also, SBIR has congressionally mandated 2.5% budget and 0.3% for STTR. These are small business and innovative research award. These are technology development, but in STTR, one partner can be from academic institute, while a small business, all the partners are from a small business. So in that also metabolomics we are promoting. And they have different phases and different amounts. So if anybody has some idea, you can discuss with me during the meeting and I can tell them how to proceed for the next step. Then we have one program, it is called IMAT, Innovative Molecular Analysis Technology. This is based on 3 RFAs, and RFAs are such announcement or funding opportunities where certain amount like 12 million or 15 million are dedicated to that area. So this is to develop new technology or to improve sample preparation or sample processing. So that kind of award is very useful. And these are some of the examples which were, in fact, Ambion, Illumina, and Invitrogen, they got their first award through this IMAT program. And now everybody use Illumina or Affymetrix. We have one CA hub also, biospecimen resource. So those investigators who are doing on cell line or in animal models and they don't have access to human samples, then also we can help them to get those samples or do that research. In our program, we support a lot of cohorts. And these are like nurses' health studies, physicians' health study, or women's health study, in which hundreds and thousands of people are enrolled, and they are, when they are enrolled, they don't have the disease. But every year or 2 years, we follow up them and collect the samples. So if you get some marker, disease marker in a patient, then in these samples, you can go back and see when you saw the first sign. So to identify new markers, these are good resources. So this is for biospecimen, and they are very well characterized specimen, high quality specimen. And then we are doing partnership with other agencies also. And those agencies like Wellcome Trust or Grant Challenges in Global Health or Bill and Melinda Gates. So with that also, we collaborate a lot. And then interagency agreements are also there. Like I was talking to a person from NASA. So with FDA or CDC, we have interagency agreement. NIH can work with them. So this is based on FDA, because whatever drugs or suppose metabolomic markers or in some pathway, you get some target which is associated with some disease diagnosis or prognosis, how to bring that from the lab to FDA approval. In that we can help. And this was the announcement of Translational Research Institute. So that new institute was initiated for translation research. And these are some of the examples which were used in another program. It's called Thought-Provoking Questions. So NIH, it was started. If you have some idea, you can submit to NIH at some specific place, and then projects will be funded based on that. So at the end, I would like to say that you should visit Metabolomic Common Funds. If you go to NIH webpage, you Google for NIH and then type Common Fund Metabolomics, it will take you there. So time to time we update that, and then contact people are there, so that I think will be very useful. And now I would like to take any questions that you have.